How is DNA used as blueprints in our body, and what happens when those instructions are messed up? That's what we'll be talking about in this module. This information comes from Chapter 9 in our textbook. For this module, the goal is for learners to be able to recognize the roles of DNA, RNA, and protein in our cells, to describe the steps of the central dogma of genetics, and to identify the impacts of mutations on the information system. So we'll have a total of 3 lectures in this module, the first talking about DNA, RNA, and proteins and their functions, then moving on to the central dogma of genetics, which is that flow of information from DNA to RNA to protein, and then lastly a discussion on mutations. While cells are squishy and organic, there are also ways in which they work as a computer or machine. Information can be stored within the chemicals. Chemical structures can also perform work and function like machines, building and assembling and moving material, often much larger than the molecules doing the work themselves. When it comes to cells and their structures, typically there are 2 different types of chemicals involved. There are the assembly instructions, which are the nucleic acids. These are the information storage molecules. And then there are those that are doing the job, the construction equipment, the functional components of the cell. These are the proteins. Proteins are complex and made of many parts, and it turns out that the nucleic acids are the instructions for assembling the proteins. You can think of the nucleic acids like the blueprints, and then the proteins are going to be the engines or the motors or the structures that are physically able to do the jobs within the cells. Let's talk a bit about nucleic acids. First, nucleic acids are long molecules of nucleotides. So the nucleic acids, DNA and RNA, they are the polymers, whereas the nucleotides, they are the monomers. And it turns out that there are 4 different nucleotides for each type of nucleic acid. Now in DNA, those 4 different types of nucleotides are represented by the letters A, T, C, and G. And it turns out that it's the order or sequence of these 4 letters that store information. The words, if you will, or the information in this language can be tens of thousands, even millions of letters long. Now when looking at proteins, proteins are long molecules of amino acids. So proteins or polypeptides are the polymers, and amino acids are the monomers, or the individual building blocks. But what happens with this long chain of amino acid is it ends up curling and folding on itself, forming a complex 3-dimensional shape. And it's the 3-dimensional shape of these proteins that give them their functions. And it turns out that there are 20 different amino acids that make up proteins. Proteins are assembled by structures called ribosomes within the cell. And so ribosomes are where the amino acids are linked together. Ribosomes are where the proteins are assembled. But then those proteins will be used everywhere in the cell, and sometimes even secreted from the cell. So where are these nucleic acids? Well, DNA, that stands for deoxyribonucleic acid. It's a polymer of nucleotides. DNA, the main blueprints for all of the proteins of the cell, are protected in a little bubble called the nucleus. While it is protected in there, it is also trapped in there. The DNA is not free to travel to all parts of the cell. It remains contained within the nucleus, the central structure for a eukaryotic cell. The ribosomes, on the other hand, which assemble the proteins, are found in the main area of the cell called the cytoplasm. Which is outside of the nucleus. So you can really think of the cell being divided into two compartments. We have the nucleus, which is the center, and that's where the DNA is found. And then there's the rest of the cell called the cytoplasm, and that's where the ribosomes are found. The ribosomes are where the proteins are assembled, and they need the instructions from the DNA. But the DNA cannot leave the nucleus. Here enters RNA, a small temporary copy of a specific section of the DNA. So an RNA is a molecule, a nucleic acid like DNA, but instead of being an encyclopedia, you can think of it as a photocopy of one page of the encyclopedia that can then go out and do its work and is eventually destroyed. So, here's an example to understand how DNA, RNA, and proteins all relate to each other. Imagine that there is a book that's held in the library, and one of the pages has very important information. So this book is in the library, it has important information, but this book isn't available to be checked out. Instead, it's a reference book. It needs to stay in the library. So this book is not able to leave the library, but you can read it there. And so when you're in the library, you're able to make a copy of that information that you're able to take with you. Whether it's a handwritten copy or a photocopy, you make one small temporary copy of some of that information. Now, the handwriting may be a little different. It's not exactly the same, and you certainly haven't taken it out of the book. The book is still complete with all its information. You just now have one additional small copy of it. Once you have transcribed it or written it down, you return the book to the front desk and leave, ready to go to the manufacturing facility outside of the library. Because those instructions you copied down, those are assembly instructions. Now, the manufacturing facility operates in a language different than the library. But through the process of translation, you end up with a new product in the new language. These are the steps of the central dogma of genetics. This example explains the relationship between DNA, RNA, and proteins within the cell. The DNA is in the nucleus, it's in the library, and it can't leave. mRNA can be synthesized or made in the nucleus, but then it can leave the nucleus to go to the ribosomes, where a process called translation occurs, where the information in the nucleic acid language of the DNA and the RNA is translated into a protein product, a product in a new chemical language. And so this flow of information from DNA to RNA to protein is known as the central dogma of genetics. And this flow of information, the information process is the same in all cells. So how do cells get the information to flow from DNA to RNA to protein? That's what we'll be addressing in our next video.